Hello students, welcome to KENE'S Educare, a group of KENE solutions. This is part 8 of lesson The Fundamental Unit of Life. So as you all know, in part 7, as part 7 was very small, we were not able to cover like much of the part for the lesson Fundamental Unit of Life. We just studied about the cytoplasm in part 7, but now in this part we can even complete with the cell organelles that the cells have inside them. Okay, and cell organelles are like the component of the cell. They are the part with which the cell is made up of. Okay, so you should be aware of it. And before I start with this part 8, that is with respect to the cell organelles, I want each one of you to please revise whatever you have in your, like whatever notes you have taken in your book, starting from part 1 till part 8. I hope everyone has started revising it. So I'll show you all the slides one by one, and you have to go through it and like see, revise each and every point that has been given. So we have started this lesson by discussing on cell theory, where we have discussed about Robert Hooke. So please go through the slide everyone quickly. After the cell theory, we discussed on like what exactly a cell is. So cell is a Latin word for little room, and it is the basic structural and functional unit of life, right? So I just want every one of you to please go through it. After this, we discussed on like what are living organisms made up of. So for that, we studied on like an activity 5.1, where we were using onion peel, and like we observed the onion peel under the microscope, under a compound microscope, and these are the structures that we were able to observe it, right? So I want you all to please go through it once, everyone. After this, we discussed on like this activity 5.2, where we were discussing about the shape and the size, like how exactly the cells are, what are the difference between them, and now you should be able to answer each and everything that we studied so far. Then we discussed on this different type of cells which are present in our body. So I want each one of you to please go through it. Then we discussed more about the division of labor, and I want you all to please go through it quickly. Then we discussed on like what is a cell exactly made up of and what is the structural organization of a cell. So I want you all to please go through it. Then we discussed on the cell membrane, which is also called as plasma membrane in detail. And here we studied about diffusion and osmosis, which is again very important. So I just want you all to please go through it quickly. Then here we have like the exact information on diffusion and osmosis which has been given. So please go through this slide as this slide is very important. And again the next slide where we have the information on hypertonic solution and isotonic solution. That is also very important. I hope everyone is going through it. Then here we have the another important slide that is about all the three types of solutions that we have. I hope everyone is going through this slide as well. Okay, so this is how you have to draw hypertonic, hypotonic, and isotonic solution. Okay, you should be knowing how exactly we need to do it. Okay. Then after this we discussed on this activity 5.3, and I hope every one of you remember when I tell you to revise this, like revise the activity, it means you have to tell the observation, like the procedure that you're going to follow, the observation, what are you going to observe, and lastly, the result that you can come up with. Okay, so you have to revise the activity as such, and I hope every one of you have started revising it as well. After this, like we discussed on this activity 5.4 as well. So here, while while we go through the activity of raising, like once we are going to put water in the raising, you'll find out that the raisins get swollen, and why the raisins they swell up, or the size of the raisins increase, it is because the water from outside, the water from the beaker has entered inside the raisin because of the osmosis process, right? Diffusion. So you can say that diffusion is important in exchange of gases and water in the life of a cell. So this activity is very important. So over here you should be knowing the procedure, the observation, and the result. Okay, so instead of going through the screen as you're revising it for like uh, two through three third time this time, so make sure that you're able to explain the procedure, observation, and the conclusion. Okay, so like we had one more activity, we'll discuss that as well, but before that please go through it once everyone. Here, like this is one of the best example of diffusion, and in this case, because the water content from outside has entered inside the resin, it's it's a kind of hypotonic solution, right? After this, like activity 5.4, here we have a concept called as endocytosis in detail. Now, what happens in endocytosis is, like amoeba is one of the best example of studying endocytosis, as we know that amoeba doesn't have any shape and it is made up of a single cell. Amoeba is a unicellular organism, right? The organism which is made up of only one cell, they're called as unicellular organism, and amoeba is one such example. So amoeba, it engulfs the food from outside, okay, from its external environment, and this process of obtaining the food, we call it as endocytosis. Okay, so like you have to, I hope everyone has taken the definition of endocytosis. So please go through the slide as well. After this, we discussed on cell wall. So I want each one of you to please go through this slide and always remember one thing, that animals do not have cell wall, whereas plants have it, right? So I just want each one of you to please go through it once. I hope everyone is reading it, and here we have another process known as plasmolysis, which has been explained. Okay. I hope everyone is going through it and even go through the structure that has been shown on screen. Then we discussed on activity 5.6. So I just want each one of you to please go through it quickly. I hope you all are going through this figure. This structure is going to help us a lot in understanding what is the exact activity that we have performed. After this, we discussed on nucleus in the last class. If you all remember, so I want you all to please go through this diagram as well as the information that has been given. I hope everyone is going to this slide. After this, we discussed more about nucleus, about the two layers that the nucleus have, that is the outer and the inner, right? So I want you all to please go through it properly. So sorry. Wait. Then here we have information on prokaryotic cell. So I just want you all to please go through it properly. Then after that we discussed on cytoplasm, like what exactly cytoplasm is, and we did that in the last class, right? So I just want each one of you to please go through this slide, because today we'll be starting and learning about the cell organelles. After this, like here we have some more information about the virus which has been provided. So I want you all to please go through it quickly. After this, now we need to start with the cell organelle part. So see, every cell has a membrane around it to keep its own content separate from the external environment. Okay, now we also want our own house, so that we are like away from each other. We need to like, we like to mix with the external environment, but it's not always right. We need a personal space as well. So all the components inside the cell, it's the same with them as well. So they also keep the content separate from the external, like this cell. They also have a membrane around them, because of which they try to keep the content of the cell separate from the external environment. So large and complex cells, including cell from multicellular organisms, they need a lot of chemical activities to support their complicated structure and function. Now because the type of function that they are performing, the structure that they have, they are all like they have to perform a tedious work, right? So and they are kind of multicellular organism. Now what are multicellular organism? The organisms which are made up of many cells. So this multicellular organisms, they need a lot of chemical activities so that they can support the complicated structure and the complicated function which has been like provided by the cell. So to keep these activities of different kinds separate from each other, the cells use membrane bound little structure, which we call it as organelles within themselves. Now as I told you that they all are separate from each other, right? And that is so because they use membrane bound cell organelles, like they have this membrane bound cell organelles which are present within them. And this is one of the feature of eukaryotic cell that distinguish them from prokaryotic cell. Now if you remember, we have discussed this before as well, that prokaryotic cell, they do not have well defined nucleus, whereas eukaryotic cell, they have well defined nucleus. And there is one more difference, that prokaryotic cells do not have membrane bound cell organelles, whereas eukaryotic cells, they have membrane bound cell organelles present in them, right? So this is one of the features of eukaryotic cell that distinguish them from prokaryotic cell. So some of these organelles are visible only with an electron microscope. It's not that you can like all the organelles of the cell are visible, like our hands, our legs are visible to us. It's not the same with the cell. Even cell is very microscopic, right? If you have to observe the cell, you have to observe it under a microscope, and if you have to observe the organelles of the cell, for that you will need a compound microscope. Okay, because like the organelles of the cells will not be visible that properly. So we have talked about the nucleus in previous session. So we have some important examples of cell organelles which we will discuss now. And this important organelles of the cells include endoplasmic reticulum. So endoplasmic reticulum is one of the organelle. Then we have Golgi apparatus. Now always remember, when it comes to endoplasmic reticulum, the cell has two types of endoplasmic reticulum in them, which we will be discussing now. So the first type of endoplasmic reticulum is known as smooth endoplasmic reticulum, and the other one is rough endoplasmic reticulum. Okay. So we have endoplasmic reticulum, we have Golgi apparatus, lysosomes, mitochondria, and plastids, and even there are vacuoles which we are going to discuss about. So we are going to study on this 5 to 6 important, you can say, organelles which are present in the cell, and they are important because they carry out some very crucial functions in the cell. We are studying them for a reason, and what is the reason? They perform some of the crucial functions in the cell, and for that it's very important that you are aware of each and every organelle present in the cell. Okay, now I want each one of you to write some essential points from the screen and even draw the structure of the cell that has been given on screen, because that is very important again. Okay, so I want you all to please do it quickly. I hope everyone is going through it. I hope everyone is drawing the structure figure and writing certain points from here, because after that we will be starting with the endoplasmic reticulum and we will be studying the two types of endoplasmic reticulum in detail. Okay, so now we can start with endoplasmic reticulum, which is the cell organelle. So see, endoplasmic reticulum, which we write in short as ER, is a large network of membrane-bound tubes and sheets. What is ER? ER is a large network of membrane-bound tubes and sheets, and it looks like long tubules or round or oblong bags, which we call it as vesicles. What does it looks like? It looks like long tubules or round or oblong bags as vesicles. So you can see, like, you might have located the two types of endoplasmic reticulum over here. One is the smooth endoplasmic reticulum, which is brown in color, and the other one is rough endoplasmic reticulum, which is yellow in color, right? So the ER membrane is similar in structure to the plasma membrane. Now we know plasma membrane is also called as cell membrane. So the ER membrane is similar in structure to this plasma membrane, and we have two types of ER. One is rough endoplasmic reticulum, and which we write it as RER, and the other one is smooth endoplasmic reticulum, that we write it as SER. Okay, so RER looks round under a microscope because it has particles called ribosomes which are attached to its surface. How RER looks like? It is going to look round under a microscope, and the reason behind that, like reason behind it looking round, is because it has particles called ribosomes which are attached to it. Okay, so ribosomes which are present in All active cells are the site of protein manufacture. What are they? They are site of protein manufacture. They are going to protein. Ribosomes are known as site for protein manufacture because they are going to help in manufacturing the protein. So the manufactured proteins are sent to various places in the cell depending on need using the ER. Okay, where will it go? It will reach different, you can say, various places in the cell, and that is all going to depend upon the need using ER. So the SER, which is called as smooth endoplasmic reticulum, it helps in the manufacture of fat, okay, and lipid, which are important for the cell function. So smooth endoplasmic reticulum is going to help in manufacturing the fat molecule, lipid molecule. Now we know lipid and fat, they both are very important for the cell, right? So they help us in that. And then some of these proteins and lipids, they help in building the cell membrane. How do they help us? They help us in building the cell membrane, and this process we know it as membrane biogenesis. Now remember one thing, the process of membrane biogenesis is very important. Okay, so you can get a question on to write an answer on what is membrane biogenesis. So you have to explain this over there. So some other proteins and lipids, they function as enzyme and hormones, and although the ER, now ER is endoplasmic reticulum, although the ER varies greatly in appearance in different cell, it always form a network system. So ER is like a long network system that we have, and thus one function of endoplasmic reticulum is to serve as a channel for the transport of material, and the material especially over here are protein, because this endoplasmic reticulum, they have ribosomes on them, and as we just discussed that ribosomes are the site of protein synthesis, right? So they have been found between the region of cytoplasm or between the cytoplasm and the nucleus. So the ER also functions as a cytoplasmic framework which provides a surface for some of the biochemical activities of the cell. So when it comes to the liver cell of the group of animals, we call it as vertebrates, and over here the smooth endoplasmic reticulum, it plays a crucial role in detoxifying the poisons and the drug. Okay, what the smooth endoplasmic does, it plays a very important role for detoxifying the poisons and the drugs which are present. Okay, Okay, so I hope every one of you are writing all the important points about ER that has been given over here on screen. I hope everyone is writing all the important points from screen. I hope everyone is writing all the essential points. After this, now we have to discuss on animal cell and plant cell in detail. So like these are the structures which have been given. I'll tell you some of the differentiating point between animal and plant cell. Okay, so now you have to look at the structure very properly. First of all, just I'll give you 1 minute, just go through both the structure properly, and then we can discuss further. Okay, so there are 10-12 points that we will be discussing, and I want you all to please write that in your book as well, because unless and until you're not taking a note of it, you won't remember it further. Okay. So see, plant, when it comes to the shape of the cell, you can see plant cell is square or rectangular in shape, whereas animal cells, they are going to be irregular or round in shape. Okay, what is the shape of the plant and the animal cell? So plant cell is like square or rectangular in shape, and the animal cell, it can be irregular or it can be round in shape. Okay, then when it comes to cell wall, now we have already studied that plants will have cell wall whereas animals will not have cell wall. If you can see all the labelings, you can find a cell wall in plant, but it is present in the animal cell. Okay, so please take these 2 points in your book first, and then we can proceed further. First with respect to the shape of the cell, and second with respect to the cell wall. So plants have cell wall, animals do not have cell wall. Then after writing about the cell wall, the next thing that we can discuss is with respect to plasma membrane. Now you can see that both plant cell and animal cells, both the cells have plasma membrane present in them. Got it? So the shape in plant, the plant is going to have square or rectangular shape. Animal is going to have irregular or round shape. Cell wall is present in plant, it is absent in animal. Then cell membrane, that is also called as plasma membrane, is present in both plant cell as well as animal cell. Okay. Then when it comes to endoplasmic reticulum, see, I'll tell you one thing, I'll tell you all the points properly. Plasma membrane, then endoplasmic reticulum, Golgi apparatus, cytoplasm, ribosomes, plastids, no, ribosomes, they are present in all. I'll repeat again. Plasma membrane, endoplasmic reticulum, Golgi apparatus, cytoplasm, ribosomes. These are 5 components of the cell which are present in both animal and plant cell. So I want you all to write this thing in your book, but please be quick with it, everyone. I hope everyone is writing all the components that I told you right now. Now after that, when it comes to nucleus, like in plant cell, the nucleus is present and it is going to lie at one side of the cell. Whereas in animal cell, you can see that it lies at the center of the cell, right? So we are writing about the position of the nucleus in plant cell. It lies at one side of the cell, whereas in animal cell, it lies in the center of the cell. Okay? And we are talking about the nucleus. Then when it comes to lysosomes, in plants, lysosomes are present, but they are found very rare. Whereas in animals, lysosomes will be present. Now you'll be studying about lysosomes and you'll come to know that lysosomes are called as suicidal bags because they are going to burst open everything. So you will get to know all the functions of lysosomes in detail. So in plant cell, lysosomes are rare, but in animal cells they are present. Okay. So please write this point with respect to nucleus and other one with respect to lysosomes. And then we have one point on plastid, vacuole, mitochondria and mode of nutrition to discuss. I hope everyone is writing about the nucleus and the lysosomes. After the nucleus and lysosomes, we can write the next point with respect to plastids. So always know plants have plastids, whereas in animals don't have plastids present in them. So plastids are absent in animal cell, whereas it is present in plant cell. Okay. Then when it comes to vacuoles, now animal cells will have small but numerous vacuoles, whereas plant cell will have large and a single centrally positioned vacuole. What this animal cell will have? They will have a single but centrally positioned vacuole present in them. Okay, so you can write about plastids and vacuoles now, and then we just need to discuss three more points, and then we are done with it. I hope everyone is writing the points with respect to plant cell and animal cell. I hope everyone has written this much. So now, like whatever points that we have discussed with respect to animal cell and plant cell, you should be knowing them all, and you have to like even draw the structure and keep in your book so that you remember like whatever points you have written that that is the similarity between plant cell and animal cell. You can write that, and the differences also you can show that. Okay. So after this, the next thing that we need to discuss is about the Golgi apparatus. So we'll discuss about Golgi apparatus, but we will be doing it further, okay, in the next part. But I hope whatever we discussed about endoplasmic reticulum, about the cell organelles, every one of you are clear with it. So we studied about the cell organelle part. We discussed with respect to endoplasmic reticulum, where we have two different types of endoplasmic reticulum, that is smooth endoplasmic reticulum, and the other one is rough endoplasmic reticulum. Then after that we discussed on the difference and similarities between the plant cell and the animal cell. So till here everyone should be aware of, okay, because like in the next class we will be starting with Golgi apparatus in detail, and what is the function, how is the structure of the Golgi apparatus. Along with that we will be discussing in detail who exactly discovered Golgi apparatus. Okay, so we'll be studying about that in detail. That is about Camillo Golgi. Then after Golgi, we'll be discussing on lysosomes. As I told you that lysosomes are called as suicide bags of the cell. So we will be finding out the reason why do we call this lysosomes as suicide bags of the cell. We'll come up with the reason for it. Then we'll discuss about mitochondria, like what is mitochondria, what is the function of mitochondria, and you should be knowing that mitochondria is called as the powerhouse of the cell, which is very important. So we'll be discussing about lysosomes, about mitochondria, and then we will be even discussing on plastids. Now we just discussed that plastids are present in plant cell and they are absent in animal cell. So we'll be discussing about plastids in detail as well. And after plastids, we'll be discussing about vacuoles. Okay, we just now discussed that animals they have only few vacuoles, whereas plants have a centralized larger vacuole which has been present, right? So we'll discuss about vacuoles, what is the function of the vacuole, everything in detail. Then we'll discuss about cell division. Now there are two important types of cell division, that is mitosis and meiosis. So mitosis and meiosis, these are two important cell division processes which goes in our body, and these are very important for the division of the cell. So we'll be studying it further, and this is going to be the last part for the lesson that we are going to discuss on. But after this we have summary discussion, like what exactly we have learned in the lesson. We'll discuss on that, and you have few of the exercise questions to discuss. So I just want each one of you to please go through all the questions that have been given on the screen. Now the first question itself that you can see here, they have asked you to compare between the plant cell and the animal cell, right? So we just now discussed about it. So I just want you all to please go through it properly. I hope everyone is going through the question that I'm showing you on slide, as we will be discussing on all the questions at the end of the lesson. I hope everyone is going through all the questions. I hope you will be able to explain at least few of the questions till now, because we are almost done with the lesson. We are only left with few part for the lesson, and you should be able to explain like few of the answers till now. So this is how we will be ending up with the lesson, the fundamental unit of life, after discussing on this 10 questions that have been given. And I hope whatever we discussed today about the cell organelles, each and every one of you are very much clear with it. Okay, now I only want one thing from you all, is like you keep on revising whatever we are studying, because for me revision plays a very important role. I have like even known it from, like, it is all from my experience that I'm stating, and I let every one of you do that, because like I don't start my part like unless and until we are not revising it, right? Because I know the importance of revision. Revision is very important for like for you understanding the concept. While you go through something for only one time, only 10% of it, but once you're going through that again and again, you know more about it. Like every time you read it, you'll get to know something new. Every time you need it, you will get to know something new about it, right? So that is why I want each one of you to please keep on revising it, because once you keep on revising it, you're adding some information to your brain to keep it stored for a longer period of time, and you even understand it well. So that is the reason I ask every time, for like I ask you to revise every time. Okay, so for today's session also, whatever we have discussed, please revise that all. And even there are few diagrams like the plant cell, animal cells, nucleus, about the endoplasmic reticulum. You have to practice all those diagrams, because this lesson is of biology, and whenever you study biology, you have to make sure wherever diagrams are applicable, you're drawing it, because biology is known for its diagram, right? So you have to do that. Okay, so I'll end the session over here, and every one of you, please make sure you all are revising it. Thank you everyone.